Marie-Caroline Daguste joined Professor Isabelle Plant's team in January 2023 for an internship initially as part of her bachelor's degree in biomedical sciences at the Université de Montréal. At that time, she began to work on endocrine disruptors, studying specifically the effect of exposure to brominated flame retardants on epithelial cells of the mammary gland. Then, having developed an interest in environmental toxicology, she decided to pursue a master's degree in the field of experimental health sciences, but still in Professor Plant's laboratory. During a master's project, she became increasingly interested in the evaluation of the effects of an occupational exposure to a mixture of EDCs on breast health. And furthermore, she's interested in social inequalities resulting from differences in the levels of exposure to these EDCs, important topic, according to women's socioeconomic characteristics. At the same time, she's involved in projects of ICEDA's or CIAPE's trainee committee. She'll talk about that later on today as well, including the Perturbation podcast that was just started. So thank you very much. Congratulations, Marie-Caroline, and the floor is all yours for the first presentation.
Thank you very much for the introduction. Can I share my screen at the same time as you, Jan? Or
let me see. I hope so. If not, let me just stop sharing here. Okay, so you should be able to do it now. My apologies.
It's okay. All right, so everybody can see my screen.
Looks great.
Perfect, thank you. All right, so I want to start by thanking ICEDA for the opportunity for me to present my research project. So indeed, today I will be talking about the endocrine disrupting chemicals we can find in cosmetic products and how they can affect the health of personal care workers. So to start, endocrine disrupting chemicals, or EDCs, are well defined in the scientific literature. Indeed, a lot of these molecules have been associated with effects on reproductive health. And one pathology that's oftentimes pointed out is breast cancer. However, what we can see is that oftentimes we are evaluating EDCs individually. But when we think of our exposure in real life, we're not exposed to one EDC at a time, but to several EDCs at the same time. And it is especially true when we think of personal care workers. So that is women who are nail technicians, so giving pedicures and manicures, women who are beauticians, so giving skincare services, and women who are hairdressers giving hair care services. Since these women are using daily cosmetic products for their work, they are probably more exposed to EDCs than the average. So this population is unequally exposed to EDCs in comparison with the average woman. And even though we know they are more exposed, there are a few studies who are evaluating the occupational exposure effects on breast cancer. Also, the measures that are in place to regulate the use of cosmetic products in the workplace are currently inadequate because they are based on these studies that are studying EDCs individually, which is not representative of the exposure of the workers. The few data that we have on cosmetic products in the literature are often based on customer exposure and not occupational exposure. An example is this study that was done in New York City, where 3,000 women who developed breast cancer were questioned on their lifestyle, the products they're using daily, and also their socio-demographic characteristics. And researchers were able to make associations between the products that these women were using and some clinical pathological features of these women's tumor. Two products came out as risk factors for breast cancer, and these products are hair dyes and relaxers. Another interesting fact that was pointed out in this study was that we saw that women that were using dye for over 10 years, they were associated with different effects in Black women and in white women. And oftentimes clinical clinical features were more severe in Black women than in white women. So by taking all of this in consideration, we can see that there is a disparity in the exposure to EDCs, a disparity that is linked to the profession of the women and also to their ethnicity. We can also see that the study of EDCs in the literature is not representative of the real exposure of women working in personal care fields. And lastly, the studies that are looking at the effects of an occupational exposure on personal care workers are not, they're not numerous in the literature. So by taking all of this in consideration, we established the hypothesis that women working in personal care workers are probably at a higher risk of developing breast cancer due to their high level of exposure to a mixture of EDC. To validate or invalidate our hypothesis, we established the following main objective, which is to assess the social inequalities and the effects of an exposure to a mixture of EDCs on the health of personal care workers. We have three specific objectives. The first one is to assess social inequalities related to EDCs exposure. The second one is to characterize the exposure to EDCs of personal care workers. And the third one is to evaluate the carcinogenic effects of exposure to a mixture of EDCs. To do so, we recruited women that work in the personal care field and also women that work in other fields to compose our control group. And we asked them to complete an online questionnaire. We then asked the volunteer women to give us urine samples that we use in collaboration with Health Canada to identify the compounds and the metabolites that were present in the samples of these women, and also to obtain representative mixtures of the exposome of the participants that we use for our laboratory analyses. First off, for our questionnaire, the questionnaire lasted for about 20 to 30 minutes. And women would question on their social demographic characteristics, their working conditions, their knowledge and attitudes about EDCs, and their health and fertility, and also their use of products on a daily basis. So the questionnaire allowed us to have a social profile of our participants, but also an exposure profile of our participants. So what products they were using and in what conditions, how long. So it was the purpose of the questionnaire. Then for the following results that I will show, they're all linked to the 29 women that gave us urine samples. Among those 29, we had 18 that were in our control group, 7 that were in our hairdressers group, and 4 that were in our beauticians group. These women had an average age of 35 to 40 years old for all groups, and most of them were white women. They all worked about 40 hours a week, and most of them had an annual income, annual family income that was of $80,000 and more. The first thing we wanted to look at was how much our participants are exposed to these products. So to do so, we asked them in a questionnaire what products they were using, and if they were using them, how often. And each frequency that was mentioned was associated to a score of exposition. The higher the frequency was, the higher the score was. For example, if we're taking participant 221, we asked her if she's using shampoo, and she answered that she uses it 3 or 4 times a week, which is associated to an exposure score of 3. We did the same thing for all the products that were mentioned, and at the end, we were able to have an individual score for this participant by adding all the exposure score of the products she is using. Oops, sorry. So we did the same thing for all the participants in all the groups. And then we were able to have an average exposure score by taking all the individual scores of participants in one group and doing an average. And we use these average to compare the expositions of participants at home. And here what we can see is that there's no differences between our groups for their exposition at home. But when we're looking at work, we can see that our hairdressers are more exposed or have higher exposition scores than our beauticians. And you'll see here that we removed our control group because it is important that we remind you that we're interested in occupational exposure. So therefore, women who are not working in a personal care field are not using products for their work. And when we combine the exposures at home and at work, we see here that we have that are two groups of interest. There's a difference here, hairdressers having higher score of exposition. And when we compare them to our control group, we also see a difference. Then we wanted to look at the 10 most used products for each group because we wanted to have an idea of where this exposition could come from for our participants. When we look at the products that are more used at home, we see that between all of the groups, we have many products who are similar. And also what's interesting is that one product among the three groups was also used at, also had a product usage percentage that was pretty much similar between the three groups. When we're looking at the products they're using at work, here we can see that they are completely different. Hairdressers are using products that are specific for their function, and it is the same thing for beauticians. And the products they have in common are more cleaning products. Another thing that's interesting here is that when we're looking at the least used products for the participants, in our hairdressers group, we see that the product usage percentage is way higher than what we can see in our beauticians group, even though they are the least used products. And when we combine again the products used at home and at work, we can see that once again, a lot of products are similar between our groups. But when we're looking at the product usage percentage, we can see that it is different between the groups, meaning that the product usage percentage coming from the work is also influences the overall use of products. Then we wanted to look at how much participants use cosmetic products in comparison to how much they're using disinfectant products, because we know that our workers are likely to use disinfectant products in their work, but also because there are also EDCs in those products. When we look at home, we see that the proportion of cleaning products and cosmetic products is pretty much the same for our hairdressers and our beauticians. But when we look at work, we can see that our beauticians use more cleaning products than our hairdressers. And once we combine the use of products at home and at work, we can see that, once again, beauticians use more cleaning products than our hairdressers. So once again, at work, the use of products at work influences the overall use of cosmetic products and cleaning products. So to summarize this part, we can see here that exposure levels to cosmetics are higher among hairdressers, and also that the most commonly used products and how often they are used can change regarding the profession that participants are doing. And also, we can see that beauticians use more disinfectant products than the other groups. Now, for the second part of the project, we want to give a special thanks to Vanessa Isabel Martinez, because she was in charge of the ultra-high pressure liquid chromatography and high-resolution mass spectrometry system, so UHPLC HRMS analysis. As mentioned earlier, the urine samples were used with UHPLC to detect the compounds and the metabolites that our participants were exposed to. To do so, urine samples were subject to a solid phase extraction. So basically, it is a process where we isolate and concentrate the compounds of interest by using solvents and evaporation techniques. And that is how we were able to obtain our urine extracts that we then use for our non-targeted analysis with UHPLC. The EDCs that were found in the urine extracts were EDCs such as bisphenol, phthalates, and paraben. And another interesting result that we saw is that we could see that concentrations of EDCs in our hairdressers and beauticians group were higher than what we could see in our control group. And some of them could go to 35 times higher. So it is an interesting result because it reflects what we saw with our questionnaires. So there are some links that we can make between what we saw in the concentration of the urine extracts and what we saw in the questionnaire. And now it would be interesting to see if we can make a link with these results and our analysis, our cellular analysis. So for the third part of the project, we wanted to look at the effects of our urine mixtures on cells. So for our laboratory analyses, we use two breast cell lines. So we use T47D cell line, which is a tumor cell line, and we use a non-tumor cell line, which is the MCF12A. And these cells were used to evaluate the effects of urine mixtures on the viability and mortality of the cells, and also to evaluate cell proliferation, because proliferation is an important characteristic of cancer. Then we wanted to evaluate the ability of the urine mixtures to activate different hormonal pathways, but to do so, we use cells coming from a specific kit. First off, the preparation of the urine mixtures. It is important to know that all participants that gave us urine samples actually gave 3 samples over the week. Indeed, we asked participants to give us a sample A on their first day of work, a sample B on their third day of work, and a sample C on their last day of work. And then all the sample A from participants of one group, so for example, all sample A coming from participants in the control group were mixed to obtain a mixture that we called A. All samples coming from same participants in one group, all samples B coming from the same participants in the control group were mixed to obtain a mixture B, and we did the same thing for the mixture C. And then we took a small volumes from those 3 mixtures to obtain a mixture that we call ABC, and this mixture corresponds to the overall exposure to the compounds. First, we wanted to do a cell viability and mortality assessment. And to do so, our cells were seated in the 96-well plates, and they were exposed to our mixtures for 72 hours. We then used fluorescent dyes to quantify the viability and mortality of the cells. The dyes that we used for this experiment, the first one was calcine that's able to grow inside the cell, and once it is inside, it is converted by the enzyme esterase into a fluorescent calcine, and the cells are then labeled in green, and those are our alive cells. The second dye that we used was etidium, and etidium is not able to grow to the cells like calcine. The only way for etidium to go inside the cell is if there's damage to the cell membrane, which indicates that the cell is dying or dead. And once it is inside the cell, it binds to the DNA, and it then becomes a fluorescent molecule, a red fluorescent molecule. So the dead cells are labeled in red. Now for this result and also the following results, you will see that we evaluated our mixtures at a concentration of 1x and 0.1x. So 1x represents the real urinary concentration of the compounds, and 0.1x is less concentrated, so it represents lower concentrations of our compounds. And you will also see here a black line. This black line represents the viability that was induced by our procedural blank. The procedural blank, which is equivalent to our vehicle control, since it is composed of residuals from the solvents used for SPE and UHPLC analyses. So we're looking at how much our mixtures are inducing more viability or less viability than what's initially induced by our procedural blanks. And for the MCF12A cell line, we saw only one mixture that had effects on the viability, and that is the ABC mixture coming from our beauticians group, who decreased the viability of the MCF12A, both at 0.1x and 1x. Then when we look at the viability of our T47D cells, we didn't see any significant effects on the viability, whether it was at 0.1x or 1x. When we're looking at the mortality on MCF12A, once again, we don't see any significant changes in mortality for both concentrations. And for T47D cell lines, we also didn't see any changes in the mortality. Then we wanted to do a proliferation assay. So what we did is our cells were seated in a 96 well plate for 72 hours. Well, actually, they were exposed to our mixtures for 72 hours after being seated. And then we followed the proliferation using the excelligence. So the excelligence is a device that allows us to see proliferation of the cells, a live proliferation of the cells, by taking cellular index measurements every 10 minutes. And at the end of our experiment, we have curves like these, and we do an analysis of the area under the curves. On the MCF12A cell line, we saw a decrease of proliferation for a lot of mixtures that were coming from our hairdressers group and beauticians group. Mostly we saw effects at 1x, but a few mixtures were also decreased the proliferation at 0.1x. And we even have one mixture from our control group that decreased proliferation as well. For our T47D cell line, we didn't see any effects on proliferation for both of our concentrations. To summarize this part, we saw that urine mixtures do not seem to have an effect on T47D cells, and we only have one mixture that was able to decrease the viability of MCF12A cell line, and that is the ABC mixtures from our beauticians group. And then we have many mixtures coming from our personal care workers that decrease the proliferation of MCF12A. The last test we did was a human estrogen receptor alpha reporter assay, because estrogen receptor is a marker of breast cancer. And basically this test, we use cells that are continuously expressing this receptor. And once the receptor is activated, it starts the transcription of the receptor, but also the reporter gene here, that's luciferase. And luciferase is an enzyme that can catalyze a reaction that produces light. And it is this light that we are quantifying with the citation 5. So the quantification of the light is directly proportional to the activity of the receptor. So here what we saw is that our urine mixtures do not seem to activate the estrogen receptor at 0.1x, because all of our mixtures were below the activation threshold of 10% that was set by the OECD, so the Organization for Economic Cooperation and Development. So this organization sets standardized guidelines to evaluate the chemicals and their potential to be endocrine disruptive chemicals, and we can see that our mixture do not attain this threshold. So to summarize everything that we just saw, we saw that both analysis coming from our questionnaire and our chemical analysis indicated that personal care workers were more exposed to cosmetic products and EDCs than our control group. And we had many mixtures coming from our personal care workers that had effects on the MCF12A cell line, mainly one that was able to decrease viability and many that decreased proliferation. But we didn't see any effects on our T47D cell lines. And lastly, we saw that urine mixture did not seem to have an agonist effect on our estrogen receptor alpha. However, it would be interesting to see if there is an antagonist effect because we saw a decrease in proliferation. And it would also be interesting to see if other markers, receptors, such as the progesterone receptor, could, if our urine mixtures could affect these receptors as well. To conclude, even though our study is a pilot study because the number of participants we have doesn't represent the whole population, we believe it could inspire innovation in risk prevention, notably with approaches that would be more specific and more representative of the exposure of our participants. And at the end, it would provide a better protection for personal care workers. I would like to thank my whole team. So obviously my PI, Isabelle Plante, the students in the lab, and also the intern. And I want to give a special thanks to Roxane Quentin, who was an intern who worked with me on this project. And thank you to the funding organisms and our collaborators. And thank you to all for listening.
Excellent. Thank you very much, Marie-Caroline, for a very interesting talk. The floor is open for questions for Marie-Caroline. Valérie?
I don't know if we can stop sharing, perhaps, so I can see Marie-Caroline. But anyway, well, thank you so much. It was so interesting. I really like when we're breaking the silo in science and like merge social science, epigenomics, yeah, I guess lab bench science as well, right? So this is a very cool project. So thanks for that. Well, I have many questions, so I will just start with, I guess, the one that is burning my lips right now. I saw that one of the products that you got in your urine is not BPA but BPS. And it's not urine, it's a pulp and paper mill, but we are seeing very similar metabolites coming from that industry, and it's not doing anything on our ER alpha cell reporter assay, but it's really affecting our androgen receptor assays, so it's anti-androgenic. So I would suggest that you look at others of those cell reporter assays, because BPS, BPA, all those, they are not just estrogenic, they have all those different effects. And yeah, so anyway, so that was one, I guess, a comment. And perhaps a question could be, why did you merge the hairdresser and the beauticians in your urine samples, I think, when you show the different contaminants? Because you took time to explain that, they have different exposure, different this and that, but then you merge them together. Yeah, I guess that could be a question.
Yeah. Well, thank you for the comment. It was in the plans to look at the androgenic receptors. So I'm looking forward for these results. And for the why we combine these, the hairdressers and the beauticians, well, it's simply because it's a preliminary analysis. So for further analysis, yeah, we're going to separate each group.
Okay, and all the EDCs that you carefully, describe, do you know where they're coming from in their exposure? Is that that's something you're gonna try to investigate, or?
That's something we're gonna try to investigate, but I think we'll go with what the literature is saying. It's telling us. We already know, that some of them are coming from the plastics that are used for the cosmetic products. Some are antimicrobial products that are used. So I think we're going to try to make associations with what the literature is telling us.
So if I'm allowed, the last question. Sorry.
Sure, go ahead.
There's always too many questions. So is it in the plan to go back to them and say, you know what, these are the 3 products, whatever, that are causing you the most harm, you know, to go back to those people and try to kind of not help, but, you know.
Yes, it is in the plan to have like different maybe conferences or just produce different papers, like posters, if I can say, flyers, to explain to our participants the results of the study without going into specifics because it is anonymous, but we can tell them that in this group and this group, we saw these products that had this and that effect. So it is in the plan, yes. Thank you.
Okay, thank you very much. Maybe one more quick question, and then in the interest of time, we'll move on. Any more questions? If not, I have a very quick one for you, Marie-Caroline.
Yes.
So you have a very nice system with 2 cells, and one is more sensitive, so the MCF12A. And you show, you know, morphological and proliferation effects, and the mixture is of course really nice. So you have samples from, you know, these real life scenarios, which is super exciting. In terms of the analysis, you identify some key chemicals like the parabens as well, and some of those metabolites. Do you know how these chemicals individually behave in your system? Are they driving maybe some of the mixture effects?
I have to admit that I don't really know. I'm sorry, I don't really know how.
No, that's fair.
Like how maybe one molecule could be responsible for like all the results that we're seeing. But I think it's definitely something that I can look up because I'm not sure.
Yeah, I'm just thinking ahead of maybe, I mean, it's always difficult in mixtures because there's often unexpected effects, but you know, if you have controls or a future student can look at individual compounds that you identify, particularly as like being tenfold elevated in one population over the other, could be really interesting to see if that's driving potentially it, and you know, for regulations and limitations might be a bit more interesting and simple. But thank you very much. That was excellent.
Thank you.